what is the difference between the pharmaceuticals in general regarding the occurrence and the impact on the environment and the cytostatic drugs? These are the drugs used for the cancer treatment. Well, regarding the routes of entry, they are the same as for all pharmaceuticals. So from the municipal wastewater treatment for hospital wastewater treatment, a little less from the pharmaceutical industry nowadays. But the main difference is why they were not addressed so far is that their concentration, expected concentrations in the environment are compared to other drugs much, much lower because the quantities used for treatment of cancer are much lower for several other common drugs like for blood pressure regulation, lipid regulations, and so on. But the main difference is the property of anti-cancer drugs because these drugs belong to a group of, we could say, one of the most dangerous compounds. Most of them are genotoxic, many of them are genotoxic. They interfere with the DNA processing, with the cell signaling systems because they are designed to kill rapidly dividing tumor cells. But the problem is that they are not selective enough, so they can affect any dividing cell, and consequently they can affect also environmental organisms, in particular when the exposures, even at low doses, are continuous. We heard about this semi-persistency of the pharmaceuticals in the environment, and this holds true also for the cytostatic drugs. So the question is, does the residues of anti-cancer drug represent environmental and human health risk? And this is the question that has been addressed in the FP7 project Cytothreat that has been financed by European Union in the FP7 program, and it is now in the third year of taking place. So our objectives are to fill in the knowledge gaps that would allow for the risk assessment of cytostatic drugs when released into the environment, because there are really no data. We don't have the data on the concentrations in the environment, and we don't have the data on the toxic effects for most of them. In particular, we don't have the data on the chronic, on the effects after chronic exposure, which is typical for the environmental contamination. But this is the problem also for many other pharmaceuticals, that many times the data, ecotoxicological data, are based on the acute toxicity test. So in this project is divided to several parts. As you can see, one important is development of the analytical methods and application to the environmental studies, because the problem is that also analytical methods are currently not very well developed for the most of the cytostatic drugs. It is the consequence, they are not there is no need to monitor them, and of course they are then not monitored, which we heard today several times. The second aim is to produce, in this first chemical analytical part of the project, we are also addressing the transformation products and metabolites. The second aim is to produce ecotoxicological data for selected cytostatics. So we had to select only a group. We selected 4 cytostatics for the whole ecotoxicological studies. And the third aim is to develop molecular biomarkers that would allow for the prediction of the long-term effects of the cytostatics and their mixtures to aquatic organisms and humans. To this end, we are applying this gene expression analysis, and we are trying to find appropriate biomarkers that would allow us for such prediction without need to conduct the chronic exposure assays. And of course to perform a risk assessment of these drugs and mixtures. So cytostatics belong to the dangerous compounds, and according to REACH regulations, but they are not the matter of the REACH regulation. They have, the pharmaceuticals have their own regulations, and the concentration, which the predicted environmental concentrations that triggers the need for the environmental risk assessment is 10 nanogram per liter. So the environmental risk assessment, according to EMA, European Medical Agency guidelines, is based on the tiered approach. In the first phase, it is necessary to estimate predicted environmental concentrations. If those are higher than 10 nanogram per liter, then second phase is necessary to be performed, which includes also the basic toxicity test. This one only. And of course, then if this ratio between predicted environmental concentrations and predicted no-effect concentrations, because these are still predicted because they are based on the acute tests using uncertainty factors, then no further action are needed. But if it is higher than this ratio, then chronic toxicity tests are needed and some other specific tests, bioaccumulation and so on. This holds true for all the pharmaceuticals, except that this trigger value, 10 nanogram per liter, does not hold in certain cases. For instance, in the case of high bioconcentration or specific toxic effects, like for endocrine disruption and also for the mutagenesis. So how to, well, actually the data, analytical data, the real occurrence, the data on real occurrence in the environment are really scarce. Actually, some cytostatics has been detected at nanogram, but some also at microgram per liter in wastewaters, but not in the river waters. Usually, so far, there are really only few reports on that. So we decided to estimate the predicted environmental concentrations based on the consumption data. We obtained this data for Slovenia from our oncological institute for 3 years, for the years 2009, 10, and 11. And we were then lucky that in 2012, a French study was published after we made all this analysis. So I will now present this consumption data and in comparison to French data, because here we can clearly see which, well, according to the mechanisms I forgot to tell, action, the cytostatics are different. The groups are anti-metabolites, then protein kinase inhibitors, alkylating agents, monoclonal antibodies, alkaloids, and so on. So this data on the consumption show that anti-metabolites, the first group, are the most consumed drugs, cytostatic drugs. And the second group are protein kinase inhibitors. This information was particularly interesting because these are very new drugs that are used in the cancer treatment. And the consumption is really increasing. Here you can see in France for the factor 2.1, but here you must take into account that the difference is between 2004 and 2008, and also in Slovenia for the fact for 30% per year. So this is interesting because we can expect increase of these drugs in the environment, because also the treatment with these drugs is different compared to other cytostatics, because they are actually used for the treatment for the long-term, long life. This is a whole life exposure treatment in the cases of this type of cancer. And another group that is also increasing very rapidly are monoclonal antibodies. This is another group that is both proteinase kinase inhibitors and monoclonal antibodies are associated with these new cancer treatment approaches based on the personalized treatment, which is associated with very good diagnostics. Otherwise, they are not useful. So these two groups are becoming of particular interest, except that maybe for monoclonals are not so important because they are degraded very rapidly and ineffective. But another thing is with protein kinase inhibitors, which are small molecules. We can also see that per capita the consumptions are quite comparable in Slovenia and in France. Also, the colleague showed that for other pharmaceuticals, there are not big difference between the different countries, even more different than France and Slovenia. Well, from this data on the consumption, we then calculated predicted environmental concentrations, and the highest, you can see that we calculated two types of predicted environmental concentrations. The conservative, where we didn't take into account any metabolism of the compound, and the refined one, where we take into account, these are the last two columns, where we took into account also the data on the metabolism. So how many unmetabolized compound is excreted, but we didn't take into account any wastewater treatment, possibility of wastewater treatment degradation in these calculations. And you can see that actually the predicted environmental concentration, the conservative ones, are higher than 10 nanogram per liter for capecitabine. Capecitabine is the anti-metabolite and is a product of 5-fluorouracil, which is the third one in this list. The second one is hydroxycarbamide, but this one is also used for other indications. And others are, the predicted environmental concentrations for others are lower than 10, but because these are the chemicals with a very specific mechanism of action, we considered that also 1 microgram per liter is the concentration of relevance. And also, if we look the groups, anti-metabolites or protein kinase inhibitors, you can see that the values are about 10 nanogram per liter, and we can actually, with the compounds with similar mechanism of action, we can also expect maybe this is one of the tasks also of the project to induce synergistic or at least additive effects. So from this data, we selected 4 cytostatics, as I mentioned, for the further studies. These are 5-fluorouracil. Here I also listed their genotoxicity data because this is important, and carcinogenicity data. So these are not the most, except for fluorouracil, the most consumed, but we decided to select from each group according to the mechanism of action 1 compound. So we have 1 anti-metabolite, fluorouracil, which is genotoxic, positive in all genotoxicity tests, and it belongs to group 3 according to classification, carcinogenicity classification by ER. So it is not classifiable as human carcinogen. Then cisplatin, it is also positive for genotoxicity data, and it is probable human carcinogen. Topoisomerase inhibitor etoposide is genotoxic and is human carcinogen, while for tyrosine kinase inhibitor imatinib mesylat, there is very, very scarce data on the genotoxicity. There is no data on carcinogenicity. There are some in vitro assays that showed positive and negative data. Well, these data are usually for the pharmaceuticals, for humans, available because toxicity data, because from the preclinical studies before the registration. So in ecotoxicities, we performed actually ecogenotoxicity. So we applied ecotoxicity test, the standard one with algae, cyanobacteria, crustacea, and fish. And in addition to perform acute and chronic toxicity test, we measured also genotoxicity endpoints in these organisms. And in addition, we included also other organisms, muscle. More data will be presented by a colleague later on. And also plants as primary producers, because it has been shown that plants might be quite sensitive to the genotoxicity and toxicity of different genotoxic agents, but cytostatics so far has not been tested yet. And as I already mentioned in the beginning, in zebrafish, in the long term, assay, 2-generation assay, I will show you later. We also did the gene expression analysis, so in order to try to find the biomarkers for the prediction of the long-term effects. So here are the results of the acute and chronic toxicity. As you saw in that, in the toxicity evaluation, if we have data on acute toxicity, usually the uncertainty factor of 1,000 is used. And here, for instance, in the case of the acute toxicity test in Crustacea, Ceridodaphnia dubia and magna, you can see how much more sensitive, how much lower are EC50 values after the chronic exposure, more than several order of tiles. While with other compounds, this difference were not so big. And etoposide, for instance, was in acute toxicity test was either non-toxic or very slightly toxic, while in chronic toxicity test in Daphnia magna, it showed quite low values. While in fish, we so far didn't perform all the tests yet, except for the toxicity test in zebrafish embryos. And you can see that the zebrafish embryos were the least sensitive compared to other tests, to the sensitivity or susceptibility of other aquatic organisms. Yeah, and then we performed the genotoxicity assays in all these organisms that are listed here. We performed DNA damage assay with the COMET assay, which measured primary DNA damage, and the chromosome and the micronucleus assay, which is another endpoint measuring chromosomal aberrations. So let me show the results. Here you can see that all 4 compounds in Daphnia magna induced DNA damage measured with the COMET assay. And what I would like to point out is for etoposide and imatinib that these effects were observed at concentrations that are even lower than EC10. This is particularly important because this means that the animals are not dying, but there is the probability of induction of mutations and then changes in reproduction and so on, and all those consequences of mutations. And another important result came from our 2-generation assay in zebrafish. Here the first generation was, the parent generation was treated 2 weeks, then the whole first generation was treated with the 3 concentration of 5-fluorouracil, 10 nanogram per liter, 1 microgram and 100 microgram. And then the COMET assay and micronucleo assay were performed in gill, liver, gonads, and blood cells. And here is the result, which actually we observed DNA damage in all organs except in gill, but it was significant only in liver. But this was the result that really surprised us, that at 10 nanogram per liter we observed clear increase in the micronuclei formation in this long-term 2-generation or 1-generation exposure assay. So, and it is also showing a clear dose response. We repeated these results because at first we couldn't believe that could be, really didn't expect so low, at so low. Now only quickly, we also performed, as I said, this microarray analysis. It is not finished yet, but what was interesting in this was that males and females responded very different regarding the number and the common differentially expressed genes. And the effects were clearly seen also at nanogram per liter concentration, and which is actually confirming the observation with the micronucleus assay. So the question is there a risk? While the PEC and PNEC values are much higher than if we consider micronuclei in zebrafish, if we consider micronuclei in zebrafish for 5FU, while other values are actually below 1. But these are genotoxic compounds, and here maybe margin of exposure is more relevant parameter. It is not always used in the environmental risk assessment, but for instance, for genotoxic compounds, for the human risk assessment, it should be 1,000 to indicate low concern. For the environment, for sure, 10,000 wouldn't hold, but it is just for the comparison. But nevertheless, margin of exposure values are quite low, except for the etoposide. So, for the conclusion, just to conclude, we saw that all 4 compounds induce DNA strand breaks, that in two-generation study, micronuclei were induced at concentration of 10 nanogram per liter, but we don't have no effect level. We should perform that way. So, the conclusion is that certain cytostatic for sure can pose threat to the aquatic organism. And the message is also that data for long-term in vivo assays focusing on specific endpoints relevant to the compound are needed to determine the significance of the presence of such pharmaceuticals in the environment. So, and finally, just a list of the contributors to this study. You can see quite a lot of us are collaborating in this project. Thank you very much for your attention.